So now we'll look at primary growth in all of the different organs of the plant. So primary growth, remember, is lengthening. The plant is getting taller. So here we have roots. We have kind of a thimble shape down here. This is a root cap. And this root cap is going to help protect the cells in the apical meristem. Remember, all primary growth is going to happen thanks to apical meristems. So here's the meristem. The meristem is where the cells are actually doing mitosis. And this needs a cap because as this is going through the soil, remember this is a root, it's going to cause damage. Apical meristems are delicate, so it needs to be protected. So the root cap actually secretes a polysaccharide slime that acts as a little bit of a lubricant to help it move through the soil. Growth occurs in 3 places. So the first thing that happens is the cells have to divide to make more cells. Then those cells get longer, and that's going to actually push the root down, is by the cells getting longer. And then eventually, once the cells have elongated, they differentiate into the 3 different types of tissues. They're either going to become dermal tissue or ground tissue or vascular tissue. So let's start down here. We've got our zone of division. So down here, the cells are actually doing mitosis. So that's kind of the portion down here, as well as including the apical meristem. Look at the cell sizes here. They're a lot smaller. Each of these little squares is smaller because they're going through division, making lots and lots of brand new, tiny little baby cells. And then from there, all the way through here, you can see step 2, the zone of elongation. So if you kind of go up this zone, you can see that the cells get longer and longer and longer. The cells aren't moving. What happens is when this cell gets longer, it pushes this down, and that's how the roots grow, is this zone of elongation. And then finally, in the zone of differentiation, the cells are going to either become an epidermis, they'll become cortex, or they'll become vascular tissue. And that's going to be a mature cell that's going to go ahead and function the way it would in a mature plant. Notice again, we have the root hairs. Those are epidermal, and they help significantly increase the surface area for absorption of water. Here's a cross-section of the roots. You've got the dicot on the left and the monocot on the right. And you can see that both of these have this endodermis, which is another layer of dermal tissue here in the center. And then it surrounds the vascular bundle. So here we've got the xylem and phloem for the monocot. I like the dicot. The xylem actually creates this little X. So the X is the xylem, which I think is super helpful for remembering it. And then the phloem is found in these little pockets in between the X arms. So they both have these little vascular cylinders. This endodermis is selectively permeable. So this is what creates a boundary between what the plant absorbs and what it does not absorb. So if the plant is going to absorb something, it has to go through the endodermis in order to get into the vascular structure and then actually get up to other parts of the plant. If a root is going to emerge as a lateral branch, it actually emerges all the way from in here from the vascular cylinder. So this is a very disruptive process. You can see that it essentially destroyed this much part of the root. So this is only going to be energetically efficient if there's a nutrient-rich resource over here that it's going to reach out for. And you can already start to see the root cap is starting to form here. We're going to see the meristem there. These cells are kind of elongating and pushing this root out sideways. Next, we have primary growth of shoots. So shoots is talking about stems and leaves. So again, we're looking at apical meristems. So here's an apical meristem, and this is protected by 2 very young leaf primordia, meaning these are going to eventually become leaves. They are not leaves yet. But this helps to protect this very delicate meristem. Notice there's no cap because these are just growing through the air. There's no actual dirt that these have to grow through, so there's no need for like a root cap on the shoots. We have axillary bud stems. These are kind of leftover pieces of meristem tissue. These have the ability to become apical meristems. So these are not going to grow tall necessarily. What these are going to lead to is things like branches. And then they're going to become apical meristems on those branches. There is a phenomenon called apical dominance. So this is my shoot apical meristem, and these are my axillary stems. The closer this distance from an axillary bud to the apical bud, this apical bud is dominant and it's active. So it actually turns these off and won't develop branches here. Further down the plant, there might be an axillary bud that's far enough away that it escapes the reach of this dominance, and it's okay to go ahead and start forming branches. Those are leaves from this branch. So one thing you can do when you're like trying to shape a bush or make it look the way you want it, usually people want bushes to look very full of branches. So in order to do that, you need to prune these off. If you cut off the tips of the tops and the top apical meristems, that's going to allow these lateral meristems to become uninhibited and go ahead and grow if this is cut off and this dominance disappears. The stems, these vascular tissue is laid out a little bit differently. So in here we have the dicot again on the left and the monocot over here on the right. So the monocot, the vascular bundles are kind of randomly dispersed. They're more dense over here by the edges and kind of less dense in the center. This is the dicot. You've got a vascular bundle here, here, so they form a ring around the center. The lateral shoots, so branches, right? They don't come from these vascular bundles. They come from the axillary buds on the surface, so they don't have to puncture through any tissue or cause damage to create a branch off of the stem. And then the leaf, they pretty much only do primary growth. So once a leaf is formed, it's not really going to do secondary growth at all. And inside of a leaf, this is going to develop from those leaf primordia. We have the epidermis up here and down here. So you can see that's going to include the outer layer of cells as well as this waxy cuticle on the top and the bottom. The ground tissue you can see in yellow here is these mesophyll. So these are responsible for doing photosynthesis, as well as the bundle sheath cells here. So the bundle sheath is going to surround the vascular tissue, and that helps to protect the vascular tissue. And then again, purple, we have vascular tissue, we have the xylem and the phloem. So this is going to allow water to come to the leaf and allow the leaf to carry photosynthetic products back to the stem or roots or wherever in the plant needs it.